Hi, this is Dr. Launen, and I am posting this to wrap up our molecular biology bootcamp topic so that when we meet next, we can move on into some more detail about finding genes, looking for open reading frames. So in class, we covered the structure and function of proteins, and we also covered the structure and function of DNA. And we ended by talking about the fact that DNA is found in chromosomes organized into a structure called the double helix, and that we'll really think about a chromosome as a long string of DNA nucleotides in this genomics class. So before we move on, let's define the word gene. So we're going to define the word gene as a sequence of DNA that encodes a unique RNA molecule. Many genes are coding, which means that they're used to make proteins. However, others simply encode what is called functional RNA and are not translated into proteins. And we'll talk more about that as we look at the categories of RNA that can exist in a moment. Now, having covered the topics of protein and DNA, I next want to talk a little bit more about RNA before we put all these things together into the process of gene expression. So RNA is shown here on the left, or at least aspects are shown on the left. Here is a single strand of RNA, and it's contrasted with the double-stranded, double helical structure of DNA that we've just discussed. Now, that's a difference. RNA tends to be found in cells as single strands, DNA as double strands. Another difference is in the nature of the nucleotides that make up RNA versus DNA. So RNA nucleotides are generally shaped the same, but there is some variety in the nitrogenous bases amongst other chemical differences. Like DNA, RNA will have the nitrogenous bases cytosine, adenine, and guanine. However, RNA will have the nitrogenous base uracil instead of the nitrogenous base thymine, which is found exclusively in DNA. So there's some chemical differences in the nucleotides that make up RNA versus DNA. They're subtle, but they're different. That's a little bit about the structure of RNA. Now let's think about the types of RNA and their functions. For our purposes, we will just consider the three major types of RNA, and they are mRNA or messenger RNA. This is the RNA that carries the information about the peptide from the gene to the ribosome. So this is an informational, short-lived molecule, just flops around in its single strand. It will be used by ribosomes in order to produce a peptide. So what's in this messenger RNA is telling the ribosome which amino acids to place into a new peptide that it is making. This is coding RNA. The second category of RNA, which is considered a functional RNA, is here. This is transfer RNA, or tRNA for short. The job of transfer RNA molecules is to carry amino acids one at a time into ribosomes so that they can be incorporated in new growing proteins. Here is a ribosome. It's a little hamburger-like structure in a cell made of a mixture of proteins and an additional category of functional RNA called rRNA or ribosomal RNA. The ribosomes are the structures that actually make proteins. They themselves are made out of protein plus rRNA. So there are three categories of RNA shown there. Two are structural, ribosomal, and transfer, and one is informational, messenger RNA. All types of RNA are made by a process called transcription, which is accomplished by the enzyme RNA polymerase. All right, so we've talked about DNA, we've talked about RNA, and we've talked about proteins. We've also introduced the words genotype and phenotype. Now let's put these ideas together into gene expression, or what is referred to as the central dogma of biology. Here, that means using a gene of DNA to produce messenger RNA, which will then be used to produce a peptide, which will fold and become a protein, giving an organism its phenotype. So the conversion of DNA to RNA is called transcription, and the conversion of RNA to protein is called translation. So genes are transcribed, RNA is translated. Transcription is done by RNA polymerase, a protein enzyme. Translation is done by structures called the ribosome or ribosomes. So let's think back to the decoder ring. Okay, so if we think about that, one of the things that we should acknowledge, see if this works here, is the base pairing rules. So in the process of gene expression, let's see, whoops, let's go back, lost control over my mouse, here we go. Let's imagine that we have a sequence of DNA. Let's say the DNA equals A, T, G, 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 C, C, okay? And we're saying, okay, we want to transcribe that gene into RNA. Well, then our RNA would be, where there's an A, there won't be a T because there are no T's in RNA. There will be a U. So it's A to U base pairing when we go DNA to RNA. Where there's a T, we'll have an A of RNA. Where there's a G, we'll have a C, another C, another C. Where there's a G or a C, we'll have a G, and a G. It's a one-way process. As we go from DNA to RNA, that is how we would transcribe that information. It's similar to the DNA base pairing rules we see in the double-stranded DNA molecule, but just a little bit different in that there's no T, instead there is U or uracil. Okay, so that's how transcription would work. Now, how would translation work? So in translation, base pairing also plays a role, but I'm not going to get into that in this lecture. Instead, I'm just going to focus on a simplification at the level of decoder ring. The ribosome is essentially utilizing the codon table, only it's doing it with chemistry instead of reading. So the ribosome is going to look, and if it sees a stretch of U's, U, then it's smart enough to know that it should incorporate the amino acid phenylalanine. If it sees CUU, it will incorporate the amino acid leucine. If it sees AAU, it will incorporate the amino acid asparagine, and so on. And that is how translation works, and we're going to leave it at that level for this class. So what I'd like you to do is two things. I would like you to review this practice activity using the codon table and using the DNA to RNA base pairing rules that I told you about. And then I would like you to do this in your notes, and we'll check it together as a class. So do part B in your notes, okay? And then, so that's doing this work, right? And then the second thing is I would like you to complete and upload the practice sheet that is located on my website in the area called Molecular Biology Bootcamp. And I'd like you to do that and upload it before the next class. I'll send you an email to remind you about it. And for now, that's it, folks. I will see you soon in class.